Is there a more famous image among bio students everywhere than an image of the cell? Probably not. Today we're going to be going over the anatomy of the animal cell, a type of eukaryotic cell. I'll also be doing this for the anatomy of the plant cell as well as the anatomy of the prokaryotic cell. I'm going to be filling out this worksheet that I've created, and if you'd like a copy of this, I will have a pinned comment below. On the bottom right, we have a word bank that I'm going to be using to populate those gray tabs that are surrounding the cell. Without further ado, let's go. I think we have to start at the center of it all, which would be the nucleus. I'm sure we all know where the nucleus is, and here we have a couple components that we're going to be speaking about. Let's talk about the most important part of the nucleus, which would, I think, be the DNA. Now, we know that DNA is what's carrying our genetic code. It uses the base pairs A, T, C, and G. You've probably seen DNA in a couple of forms. Here we see it in an open form. So this DNA is unraveled. This DNA is able to be read so that we can express genes and proteins. You will not see the DNA looking like this if the cell is going through cell division. Instead, you're going to see DNA looking a little bit more like this. Right? See, the DNA is all bound up here in this chromosome. This is tightly wound. Transcription machinery is not going to be able to get onto the DNA and read any of it and make an RNA transcript, so we won't be able to make any proteins off of this. However, here we see the DNA open, and this form of DNA is called chromatin, and chromatin is going to be DNA plus protein. This is just DNA and proteins that are attached to it, often histones. In the center of the nucleus, we see another special organelle. This is called the nucleolus. Now, the nucleolus is known for one major thing, which is making rRNA. There's a couple major types of RNA that we learn about in this course: mRNA, tRNA, and rRNA. R stands for ribosomal. Ribosomes, you probably have heard of ribosomes. We're going to get to them in a moment. This is a ribosome. There's two subunits here. I know it just looks like a blob, but this is a ribosome. Ribosomes are made of protein and rRNA. Again, the rRNA, which is made right here in the nucleolus. So you can write here rRNA. Surrounding the nucleus, so I'll put here, this whole thing is the nucleus. Surrounding the nucleus is a special type of enclosure called the nuclear envelope. The nuclear envelope is a double membraned enclosure. Remember, the DNA is a very special component of the cell. It needs double the protection. So we have an inner layer and then an outer layer, and this is the nuclear envelope. The nuclear envelope is perforated by nuclear pores. The nuclear pores are how things get into and out of the nucleus. The nuclear pores are going to allow that. Directly continuous with the nucleus, and that might even be a question that you see, what organelle is continuous with the nucleus, is the rough endoplasmic reticulum. Now there's two types of endoplasmic reticulum. We have rough here, and we'll see we have smooth over here. Now what makes this rough versus smooth? Rough endoplasmic reticulum has these little red dots all around it, which is the ribosome. This is the ribosome that we were just looking at. So here we have ribosome. We can see that ribosomes are in two different places in the cell. We have free-floating ribosomes that are just around in the cytoplasm, and we have ribosomes which are bound to the endoplasmic reticulum. These are tethered. Now, why would we want to tether some ribosomes? Well, let's think about the process of gene expression. Genes are located on the DNA. It gets read, and what do we make? We make mRNA, right? Here's the best picture I could find. Here's DNA, and we have RNA next to it. So we're making mRNA. That mRNA wants to get to these ribosomes. They need to. The ribosomes are what are going to read that mRNA and build what? They're going to be building the proteins. So it's great that this rough ER is continuous with the nucleus, because that mRNA can exit the pore and enter into the rough endoplasmic reticulum. The rough endoplasmic reticulum is known for making proteins, processing and making proteins. These are continuous tunnels and sacs here. So that mRNA is going to be going through and exit the rough endoplasmic reticulum, and as that's occurring, it's being read by the ribosomes. Oftentimes, it's going to be fed right back into the rough ER, because there could be other post-translational modifications that can occur in the rough ER. But note that these are known for making proteins because the ribosomes are there. Now, what about the other endoplasmic reticulum, the smooth ER? Smooth ER has its own features. It's known for synthesis of lipids. This is a cholesterol molecule, which is a type of lipid. Smooth ER might be in cells that are creating cholesterol-based hormones like estrogen or testosterone. It's known for metabolizing carbohydrates. We all love some carbohydrates. It's known for detox at the cellular level. Can you think of which organs would have a large amount of smooth ER for helping with detox? The liver, right? The liver is known as an organ of detox. So of course that liver is going to have cellular components that help with that. So that's going to be the smooth ER. And finally, it's known for storage of calcium ions. If you've made it through anatomy and you've studied a little bit about muscle cells, then you've heard of the sarcoplasmic reticulum, which is a type of smooth ER. And the sarcoplasmic reticulum stores a ton of calcium because calcium is dumped out whenever we want to contract our muscle. So it's stored in that specialized type of smooth ER. So the smooth ER has those 4 major functionalities: synthesis of lipids, metabolizing of carbohydrates, detox of poisons, and storage of calcium. Okay, but back to our mRNA who went through and found the ribosome, was translated into a protein. Now what? Well, the next step is going to be towards this guy, the Golgi or Golgi apparatus. The Golgi's job is to grab that polypeptide that was made in the rough ER, and it's going to edit it a little bit more. It's going to put some lipids on it or some carbohydrates on it or maybe even other proteins on it, because the Golgi apparatus is filled with enzymes, different specific enzymes. Here's a 3D enzyme. Remember, enzymes, form equals function. If they're not in the right form, then they're not going to be in the right function. So these sacs are all filled of specific enzymes. A product is going to be sent from the rough ER over to the Golgi apparatus, and it's going to move from one to the other, to the opposite end of the Golgi apparatus. And then those products are going to be packaged. They're going to be packaged into vesicles. Vesicles are just little bubbles that hold specific products. And what's surrounding those bubbles? Well, it's the same thing that's surrounding all of the other organelles and the cell itself. This is going to be none other than the phospholipid bilayer. So note that the same thing that's surrounding the Golgi apparatus is surrounding nuclear envelope and the rough ER and the smooth ER and the cell itself, the plasma membrane. It's just phospholipid bilayer. That makes it easy when we need to package something from this organelle and send it over to this organelle, and then this organelle is going to then package it, and where is it going to go? Well, if it's packaged, it's got 2 places. It's either going to be secreted outside of the cell. Secrete means to remove, to send it outside of the cell, or it's used for itself, so it'll go somewhere inside the cell. So if it's going to be secreted, it's going to be sent to the plasma membrane. So we can see that over here. We see that there is a secretory vesicle that is being formed. Actually, I would say that this is taking in stuff from the outside, pinching off, and it's going to move inside the cell, but it can go in the opposite way too. This plasma membrane just becomes part of this plasma membrane and dumps all of the stuff outside of the cell. So the Golgi apparatus is a bunch of enzymes, and it's going to be modifying our product with some post-translational modifications. It's going to package them in vesicles, and it's going to send it where it needs to go. I can name this a Golgi vesicle. Okay, we've mentioned this guy a few times, so might as well just fill this in. Of course, this is all the cytoplasm, all this light yellow color. Now, what is cytoplasm? Well, the actual fluid that fills up a cell is called the cytosol. And the cytosol is filled with organelles, all of these that we're talking about, the ribosomes and all of these that we haven't mentioned. Cytosol plus organelles is cytoplasm. So if you see a difference, cytosol versus cytoplasm, cytosol is just the fluid. It's mostly water and proteins and lipids and whatever else, and ions, lots of different ions that are in the cell. The cytoplasm is that fluid plus all of these organelles. So that's the cytoplasm right there. And we've already mentioned the plasma membrane. So again, this is made of phospholipid bilayer. Remember that phospholipid bilayer, 2 layers of phospholipids. Remember that these are amphipathic, amphi meaning 2, right? Like an amphibian. It could be in the water, it could be on land. Amphipathic, 2 paths. It is both hydrophobic, that's going to be the inner part here, these tails, these are hydrophobic tails. That means that they're water fearing. They do not want to be anywhere near water. And these yellow bubbles are hydrophilic. You can put these around water. So the yellow bubbles, the hydrophilic heads, are going to be on the outside of the cell because our body's mostly water, so all of this is going to be water. And on the inside of the cell, this is going to be all water. Whereas the interior part here, that's the hydrophobic part. And why is this important? Well, the plasma membrane's job is to let things that are supposed to come in and keep things that are not out. And how are we going to do that? Look at this double defense. We have something that's hydrophilic and we have something that's hydrophobic. Most things are not both. So if we have something that's hydrophilic, it might be able to interact here, but it'll get stopped here. If we have something that's hydrophobic, it will just get stopped here. Of course, this doesn't apply to water, which is so small and it can just slip right through. And a couple of other things like non-polar compounds like oxygen and CO2, but everything else gets stopped at the plasma membrane. Okay, next organelle, we have another one that looks like a vesicle, and again it's very similar because it is going to be surrounded by plasma membrane. This is a lysosome. Lys means to burst or to break, and some means body. So this is just a body, and don't think of the body like our body. Think of it like a body of water. It's an entity. So to lys means to burst. So what does that have to do with a cell? Well, this organelle's job is to take in macromolecules and break them down. This is like the cell's stomach. It's full of enzymes just like our stomach is. And what do? We take in proteins, we take in fats, we take in carbohydrates, and we break them down. That's what the lysosome is doing. This is the stomach or the gut of the cell. This is the peroxisome. So peroxisome is an interesting one. When you think of peroxisome, think oxidation. And remember, oxidation means loss of electrons. The peroxisomes are a specialized metabolic compartment, and they have enzymes that remove hydrogen atoms from different substrates. So they grab hydrogens and they transfer them onto oxygen to produce H2O2, which is hydrogen peroxide. And you may know of hydrogen peroxide, it's usually under your medicine cabinet in a brown bottle. Hydrogen peroxide itself is toxic, so the cell also has an enzyme that breaks down hydrogen peroxide into water. This guy's role is oxidation. It's breaking things down, but in a special way. Its byproduct is hydrogen peroxide. Next, we have another compartment called a vacuole. So a vacuole is formed from phagocytosis. Phago means to eat. Phagocytosis or phagocytosis is when the cell takes in some solid products and then it's going to hold it in a special compartment, and that's called a vacuole. Vacuoles are these specialized maintenance compartments. There's many different types of vacuoles. They're mostly derived from the ER and the Golgi apparatus, so they have some important roles in the endomembrane system. It's really selective in transporting solutes, so as a result, the solution that's inside of a vacuole is going to differ in composition from the cytosol, that fluid that's just in the cell. There's food vacuoles, which is formed by phagocytosis. We can have contractile vacuoles in some eukaryotes that are living in fresh water, whose job is to pump excess water out of the cell. And you might know that plants have a large central vacuole, which plays a major role in the growth of plant cells. Okay, now let's get on to probably the most boring part of cell biology, which is the cytoskeleton. And a lot of students confuse these because the words are very similar, so I understand. So there's 3 components of the cytoskeleton that we are going to learn. We have microfilaments, microtubules, and intermediate filaments. So we can see 2 of these words have filaments and 2 of them have micro, so I get why it's confusing. So we're going to start with the largest one, and we're going to go from large to small. The largest are called microtubule. Microtubule has the word tube in it, and just think tube like a big round tube. So these are the biggest, and if you were to look at this on the cellular level, it does look like a tube. It's made of a smaller protein called tubulin. So again, that's an easy one to remember. Microtubules are made of tubulin. So microtubules are just a building component that the cell uses for other things. We can see right next to the microtubule, we have another structure. This is called the centrosome. You might recognize the centrosome from the chapter on cell cycles and cell reproduction. This is where the mitotic spindle is going to spin out of when the cell is going to duplicate its DNA and divide. These go to either side of the cell. They spit out the mitotic spindle, which grabs onto the chromosomes and it pulls it apart into opposite directions. So this is made of microtubules. The two pasta-looking components of the centrosome are called centrioles. So we have two centrioles that consist of the centrosome. And where do we also see microtubules? They also make up cilia and flagella, which we do not see on this cell. Next, we have the middle-sized, which is the intermediate filament. So the intermediate filament is the pink one shown here. It's a little bit thicker and it's really strong. And the one thing that I want you to note for this is it's used for anchoring. So it's structural component that's used for anchoring. Anchoring organelles, anchoring things within the cell, things that aren't supposed to be moving around freely. And finally, we have the microfilaments. So a filament is a really small fiber, right? If you think of a filament, it's something very, very small. So same here, a microfilament is this really thin, small fiber. It's made of actin monomers. And these have a couple of important uses. If you remember microvilli, it's not pictured here, but if this cell had little finger-like projections going across the top of the cell, that would be a microvilli, and its job is to increase surface area. We have those in our enterocytes in our gut cells so that we have more surface area to absorb more nutrients. Now think about this plasma membrane, which is the consistency of a salad dressing, so it's not very sturdy. So we need something to create some sturdiness if we're going to create these finger-like projections. So that's going to be what the microfilaments do. So you're just going to align over and over pieces of that microfilament to create that structure. And similarly, we have some cells called amoeba that like to push out parts of their plasma membrane and kind of walk. It's called a pseudopod, a false foot. And in order for that, again, for the plasma membrane to have any structure to it, you're going to have something behind it. We're going to see microfilaments in that instance as well. And finally, I saved the best for last, the mitochondria. Say it with me, the powerhouse of the cell. So why is mitochondria the powerhouse of the cell? Well, it makes a very important molecule called ATP. ATP stands for adenosine triphosphate. We have an ATP here. Nitrogenous-based adenine, we have the sugar ribose, and we have 3 phosphate groups. If you want a breakdown of cellular respiration, I do have a lecture on that as well. If this is all still very confusing, I have an entire lecture on the cell. It's called Chapter 6, a cell. I'll link it here. Keep an eye out for more of these worksheets. I'm making a lot more next semester, and if you have any particular worksheets that you think would be helpful, please let me know in the comments below. Thank you so much. Best of luck, and I'll see you next lecture.